Just before a note here, the cell cycle is a dynamic process. There's not just distinct phases where the cell jumps from one to the next. It's a process. The cell's always moving. I emailed you guys and sent you a link to the V-cell video, so you might want to take a look at that before you look at this PowerPoint, or you can take a look at it afterwards. It'll show you the dynamic process of mitosis. What exactly is the cell cycle? It's the process by which the cell prepares to divide, duplicates its DNA, and then in turn divides to create new cells. The key roles of cell division include reproduction, growth, and repair. For unicellular organisms, cell division is their mode of reproduction. For example, bacteria go through binary fission where they split in half in order to create new bacterial cells. An amoeba is a single-celled eukaryote, it's a protist. They also have to use cell reproduction or cell division to reproduce because they're single-celled organisms. The only way to make more of those organisms is for a single cell to divide. In multicellular organisms, cell division allows for growth and development, and also for tissue repair. For example, if you cut yourself, you have lost cells when you cut yourself. So in order to repair that damage, your cell is going to be stimulated to undergo cell division and put more cells in place of the cells that you lost from that damage. As you grow older, you start as a little baby, of course, and when you're a little baby, you're very small, and then you grow into a larger organism, to a teenager, and then an adult. In order to grow in size, your body's actually obviously going to have to increase the number of cells that make up your body itself. So when we talk about cell division as a way to grow and develop, that's because you're obviously going to grow in size. The role of cell division in the body is to conserve genetic material from parent to daughter cell, and this is whether it's reproduction for a single-celled organism or growth and repair for a multicellular organism. The DNA that's passed down from the parent cell to the daughter cell is going to be the exact same DNA. We refer to this as genetically identical daughter cells. The daughter cells that are created from the process of cell division are going to look exactly like the parent. This occurs to conserve genetic material in an organism, in a multicellular organism, so that all of their cells have the same DNA, but also in single-celled organisms as well. The daughter cells are going to look exactly like the parent. Now, single-celled organisms have a higher rate of mutation, which we'll talk about in the future when we talk about DNA, so it's not always that the bacterial cells or the amoeba cells are going to look exactly like their parent due to mutation, but more on that later. Okay, this is just a quick diagram that shows you what happens overall in the process of cell division. So, for example, at the start, if you are an organism that has 12 chromosomes in your cells, you could say that the parent cell is going to have 12 chromosomes. Then during a phase that we call S phase, during interphase, which we'll talk more about in a second, the chromosomes duplicate themselves using DNA synthesis or DNA replication. When that occurs, those duplicated chromosomes are going to be held together as sister chromatids. But at this point, you technically have 24 chromosomes now in the cell if you started with 12 chromosomes because you've duplicated that genetic material. But then when the cell goes through the process of mitosis, those 24 chromosomes are actually going to split from one another, and 12 chromosomes that are exactly identical in type to one another are going to end up into the new daughter cells. So at the end of cell division, in the mitotic division, you end up with the same number of chromosomes as the original cell. So like I said before, cell division distributes identical cells with chromosomes to the daughter cells, and we refer to a cell's genetic material, its DNA, as its genome. The genome of prokaryotic versus eukaryotic cells can be a little different. Remember, prokaryotic cells are like single-celled organisms like bacteria that do not have a nucleus. Their genetic material, their genome, is going to exist as a single circular piece of DNA, and I'll show you more what that looks like in a second. Eukaryotes, on the other hand, are going to have more than one chromosome, and they're going to have several linear DNA molecules, linear meaning in a straight line. So what does that look like? Here's a prokaryotic cell on the left. You can see that its piece of DNA is circular. There is no start, there's no finish, it's all one piece of DNA. It's a single chromosome. In the eukaryotic cell, you can't really see the linear aspect of the DNA in this picture, but you can see that there is multiple pieces of DNA in that eukaryotic cell, and it's like a string rather than a circle. Before cell division occurs, DNA exists as chromatin. When you're talking about genetic material, there's two forms of that genetic material. It's either a chromosome or it's chromatin. Chromatin is just going to be the squiggly DNA like you saw in the last picture of the eukaryotic cell. You can't see the distinct pieces of DNA. You just see what looks like a ball of thread. This chromatin is just actually thin strands of DNA that are wrapped around histone protein molecules, which I'll show you in a second. But before the cell can actually go ahead and divide, this chromatin is going to have to condense into chromosome form, and we'll see that during the process of mitosis. So here's the eukaryotic chromosome. When you talk about the linear nature of the eukaryotic chromosome, you can now see it in that X shape right there. There's a start to the piece of DNA, there's an end to the piece of DNA. It's not a circular piece of DNA like a prokaryotic cell has. The histone proteins you can see are the purple balls on this picture, and DNA winds around those histone proteins, and then the histone proteins with the DNA wrap around itself to make those coils, and those supercoils then turn into a chromosome. We refer to the histone proteins plus the DNA wrapped around them as a nucleosome, and you can see the nucleosome at the top picture there. Now, you know a little bit about the DNA, but exactly how does the cell know how to divide? Cells are going to receive something called growth factors, and we'll talk more about growth factors in the future here when we talk about cell signaling. But basically what a growth factor is some sort of protein molecule or a signal molecule like a hormone that binds to a receptor protein on the outside of a cell that needs to divide. And the binding of that growth factor is going to stimulate the metabolic pathways necessary to stimulate cell division. So a signal is going to be sent into the cell, it's going to reach the nucleus, and the nucleus is going to know that it's time for the cell to divide. Growth factors are released when something like organ damage happens, or if you get a cut or something like that. When you need to make more cells, growth factors are going to stimulate that process. Okay, so the cell cycle is split into three sort of parts here. First, you have interphase, which is the longest phase of the cell cycle. Most of the time that a cell is metabolically active, it's spending time in interphase. Then when the growth signal my fiance is screwing with me, and it's a pain. Okay. When a growth factor is received by a cell, and the cell decides it's time to divide, the cell is going to then move past that G1 phase that you see there to the S phase, which is when DNA synthesis occurs. The cell is only going to reach S phase when it's time for a cell to divide. The cell is not going to duplicate its DNA unless it's ready to divide. Once the cell gets through interphase, and it's going to hit mitosis, and mitosis is when the nucleus divides. So that prophase, prometaphase, metaphase, anaphase, telophase, that's all done so that the nucleus can divide. So the chromosomes end up in the right spot at the end of cell division. The final part of the cell cycle is called cytokinesis. That's cytoplasm division. That's when two new cells are fully split from each other and the daughter cells are being created. Okay, a cell can go through the cell cycle in about 24 hours. Like I said before, interphase is going to account for most of the cell's life, but the mitotic phase is actually going to be about the shortest part. I believe it's about 18 hours or so to go through mitosis. Don't quote me on that. Not 18 hours. It's like an hour or so to go through mitosis. It is the shortest part of the cell cycle. Even though mitosis and cytokinesis in that last slide you saw that they were part of M phase, they're not actually the same process. Mitosis and cytokinesis are two distinct processes. Mitosis is the division of the nucleus, while cytokinesis is the division of the cytoplasm. Interphase itself is divided into three subphases. G1 phase, which is also known as the growth phase, S phase, which is a synthesis phase, and G2 phase. During G1 phase, the cell is metabolically active, meaning it's doing its normal cell job. It's respiring, it's making proteins, it's sending signals, and all that jazz. A cell that is not planning on replicating is not going to leave this part of the cell cycle. So it's always going to stay in G1. Cells that will never divide again, so cells that have become differentiated or have become their adult form with their specified task, those differentiated cells are typically going to move into what we know as G0 phase, which is also known as a resting phase. At this point of G0 phase, the cell no longer is going to divide. Some of your cells are so differentiated in your body that they physically can never divide again. They're just too specialized. Examples of cells like this are the neurons that send signals throughout your body. You guys know what those are. And also muscle cells. They are just so specialized and they have such specialized structures that it's not possible for those cells to divide again. Some cells can come out of G0 phase if they need to. For example, your liver cells. You may have heard before that the liver is the only organ in the body that can actually regenerate itself. Well, that's true. And those cells that are specialized in the liver cell, they can actually come out of G0 phase and return to the cell cycle and start to divide again if necessary. After G1 phase, you'll find the first cell cycle checkpoint. We'll talk more about those in the future. But the G1 phase checkpoint makes sure that everything's good to go for cell division. All right, during S phase, that's the second part of interphase. That's when DNA replication occurs. This is when your chromosome number is going to increase by 2. So in that first slide where I showed you the overview of the cell cycle, this is when you ended up with 24 chromosomes after the chromosomes have been duplicated. Once chromosomes become duplicated, there's 2 exact copies that are held together, and we refer to them as sister chromatids. So when you see a chromosome in an X form, that means that the chromosome has been duplicated. That means that each chromatid actually has exactly the same genetic information. And the reason the body holds them together is so that they don't get lost from one another. It just is a way for organization to occur. They are connected together at what we call as a centromere, which I'll show you in a second. And that centromere is actually formed by what we call cohesin proteins. Okay, this is a picture of a duplicated chromosome. You can see on the left there that those 2 chromatids next to each other, they're held together by that centromere, which is that middle point where it's sort of pinched in. And the centromere is actually made up of those cohesin proteins that kind of adhere those 2 sister chromatids together. During nuclear division, during mitosis, those cohesin proteins are going to break down so that the sister chromatids can separate and be distributed into the new daughter cells. Okay, the final phase of interphase is called G2 phase. This is when proteins are made that are involved in cell division. And the second checkpoint occurs here before M phase. So the cell is going to make sure that the cell is big enough to divide. It's going to make sure that the DNA has been replicated properly, and it's going to make sure that all the organelles and the proteins that are necessary for cell division are present in the cell. Once the cell has decided all these things are ready, then it's going to enter into mitosis.